Good evening, everyone. I have a question. How many of you have sent a message with a typo in it? It happens sometimes. We just feel a bit embarrassed for a bit, and then we just laugh it off and move on. Nothing big. But what if it was not in a message, but rather in a manual that used for building a human body? And that's exactly what DNA is. And tonight I will show you that for the first time in history, we learned how to not only read that manual, but also how to edit it. Welcome to the world of CRISPR, editing life like a glue dog. Copy and paste. It's one of the most simple things that we can do on a computer, and we do it on a daily basis. And strangely enough, we also, our cells and human and living organisms have been doing the exact same thing for billions and billions of years. Your cells work like an incredible copying machine, copying DNA every second. But here's the problem. Nature doesn't have spell check. And if during copying there's a mistake, it will be in our lives for the rest of, stay with us forever. And until recently, all we could do is just watch. And today, we're learning how to edit it. Imagine these two words, cat and car. Only one letter changed, but the words have completely different meanings. DNA works exactly the same way. Sometimes the difference between health and diseases is a single letter in our genetic information. One typo, that's all it takes. A mutation that can lead to diseases like sickle cell anemia or cystic fibrosis. It's wild to think that the entire difference between a healthy lifestyle and years of suffering is just a mistake that's even smaller than a text message typo. For decades, medicine could only treat the symptoms. And now we're learning how to fix it. Your genome contains more than 3 billion letters, 3 billion. And this Wovnik contains 100,000 letters. So in order for us to have as many letters, we'll have to have 30,000 of these Wovniks, which is approximately 2 football fields. Hunting down one faulty letter is like searching for a single typo hidden inside an entire university library. And yet, the stakes couldn't be higher. Scientists estimate that more than 10,000 diseases could be cured using simple gene editing. At the same time, all of us in this room are about 99.9% similar. Try to think about that. The person right next to you has different dreams, different fears, different music tastes, and all of that is just 0.1%. So where did we find the solution? In a futuristic government laboratory, NASA, or a particle accelerator? No, we found it in the bacteria used to make yogurt. One of the biggest medical revolutions of the 21st century happened because we understood how primitive bacterial defense works against viruses. Whenever a virus attacked, the bacteria would save a small piece of the viral DNA inside their own genome. It was biological most wanted list, a collection of molecular mugshots. Next time the virus appeared, it would already recognize it, cut it into pieces, and survive. When scientists finally mapped this unusual activity, they named it CRISPR. It stands for Clustered Regularly Interspaced Short Palindromic Repeats, which sounds exactly like something you'd come up at 3 a.m. and worry about the pronunciation later. But behind that terrible name was an incredible truth. Bacteria that evolved a strong immune memory billions of years, evolved billion years before humans even developed laboratories or microscopes or even computers. We just noticed the mechanism and learn how to use it. Scientists looked at the bacterial defense system and asked, can we borrow it? And the answer was yes, and everything changed. CRISPR went from being a bacterial survival check to one of the most prospering technologies in the current world. It's so powerful that its discovery earned 2 scientists a Nobel Prize in Chemistry in 2020. Jennifer Doudna and Emmanuel Charpentier had transformed an obscure bacterial defense system into a tool that could rewrite biology. And they won not for building a machine or finding a new planet, but rather for giving us molecular scissors. We went from trying to read the manual to actually learning how to control it. So how does CRISPR work? When people hear gene editing, they think of something extremely complicated, but in its origin, it's very simple. It uses 2 main components. The first is called guide RNA. Think of it as a molecular GPS. It tracks down the target and leads directly to it. The second one, which is Cas9. It's a type of an enzyme which works as scissors, and it cuts it. The cell naturally tries to repair itself, and here where biology begins, because cells really hate having holes in their DNA, and that's why they try to replace it, and sometimes even remove it completely. And when scientists provide the proper environment and proper template, the cells can use that template to rewrite the sequence more precisely and fix the problem. In other words, we're not just cutting DNA. We're guiding the cell's own repair machinery to make specific changes. Find, cut, and edit. It really is like a word processor, but for biology. So what can we do with it? There are three main areas that this technology could be useful in. First, medicine. Imagine a doctor who no longer asks, what kind of chemotherapy will we give to the client and the patient, but rather asks, what type of DNA do we have to remove? Scientists are already programming these scissors to help immune system recognize and destroy cancer cells and leave the safe ones working properly. This isn't a future concept. It's already working in clinics all around the world. And for families who are being told we can't manage the disease, but we can't fix it, finally hearing that we might fix the problem changes everything. Second, agriculture. Imagine a farmer staring at a field after a month of drought. For that farmer, CRISPR isn't some complicated technology. It's the difference between drought, between bankruptcy, and harvest. Scientists are already editing crops like wheat so they can survive through some harsh environmental problems. And the last one is research. For researchers, CRISPR is like switching from paper encyclopedia to a Google search bar. Questions about evolution that once took generations to explore can now be investigated in just months. We're not editing genes, but rather we're discovering why life works the way it is. And this is where things might become slightly uncomfortable. Because if we can do all of this, then where does treatment end and where does enhancement begin? Most of us would agree that curing a child with a genetic disease is a good thing. But what happens when we go further? What if we could enhance athletic ability, reduce the need for sleep, or even increase intelligence, designing people instead of treating them? We move on from medicine to genetical tuning. And I want to ask you something, and please be honest. If we had the ability to genetically enhance human beings, should we do it? Raise your hands if you think the answer is yes. Okay. And what's interesting that your answer might depend on who are we talking about. Yourself, your future child, a child with a deadly disease. The line moves depending on how personal the question becomes. And now, raise your hand if you think the answer is no. Okay. So look around. Even in this room, there isn't a clear answer. I think this is fascinating because 5 minutes ago, we were talking about bacteria and yogurt, and now we're debating whether humanity should redesign itself. Technology is moving incredibly fast, but questions about fairness, equality, and what it means to be human don't have easy answers. Just because we can edit the code of life doesn't automatically mean we should. For thousands of years, evolution wrote the history of our species on its own. For the first time, we have the ability to control ourselves. Then perhaps the biggest challenge of CRISPR isn't asking, can we edit genes, but rather asking, will we be wise enough to know what we should write next? Thank you.